thank you, Vern and John and Grant for inviting me here. It's been a really nice experience, I have to say. Actually, indeed, yesterday we had a lot of discussions, and I'm really impressed by the whole team that you have here and the translational research that you do here. It's on childhood diabetes. We do adult diabetes, but we have a little bit the same approach. I mean, we try to do translational studies, combining animal and human studies, and I think that's really the way to go. So it was really nice to see here, to be here. The only downside is a little bit that it's a long travel and then you have jet lag and so I woke up this morning at 2 o'clock. So if I fall asleep during my talk, wake me up. So we are located in Maastricht. And Maastricht, for those who don't know it, shame to you, you should come there. It's a small city, it's in the south of the Netherlands, and it's a little bit an old Roman city, so it's really beautiful, especially in summer, lots of terraces, you can drink beer everywhere. So that's good. We have 400 pubs or something, a population of 100,000 people, so that's not close. But anyway, let's go to the science. So I'm in the Department of Human Biology, which basically means that we do a lot of human research, and I'm a little bit the outcast in the department because I am also doing animal research. But we recently reopened our metabolic research unit at Maastricht, and I think we can say that we have very nice facilities to study human metabolism in all the details, and I will show you some of the data which we have, for example, with those so-called respiration chains. I don't know if you know them, but maybe you know metabolic cages for rats. Well, this is a metabolic cage for humans. You can look up the human, you measure the oxygen consumption and the CO2 production, and you basically can measure the energy metabolism of this person in the chamber without touching him. And I'll come back to that later. So other techniques, because I mean, if you do human research, you want to have non-invasive techniques. So what we do a lot is magnetic resonance imaging and spectroscopy, and we apply that to the heart, to the liver, to the skeletal muscle, so that we can really measure fat content in all those tissues, but also mitochondrial function or energy status of those tissues non-invasively. Well, here you see this respiration chamber. We always call it a small hotel room. Almost, it's very small, 2 by 3 meters or something. So most likely an expensive hotel room in Amsterdam or something. Very good. So there we can measure energy expenditure. We can also measure energy expenditure, of course, by a ventilated hood system, where you basically have the hood over the head and then can measure oxygen consumption, CO2 production. And of course, lately what we also have been doing a lot is focusing on brown adipose tissue. In 2009 we were one of the 3 groups that rediscovered the brown adipose tissue in humans. So we showed that basically all humans you can see some brown adipose tissue if you cool the subject down good enough. But I won't talk about brown adipose tissue today, but I will mainly focus on the skeletal muscle, and I will explain you a little bit why we have so much interest in this muscle. And one of the things is basically already depicted here, and that is the fact that the muscle can store quite a lot of fat. And we have heard about it more today, of course, the ectopic fat storage. You can visualize that by magnetic resonance spectroscopy, non-invasively, but what we also do a lot is take muscle biopsies, and then you can stain those muscle biopsies and do an all-red O staining, and you see all those little red droplets, which are basically lipid droplets. If you even zoom in a little bit further and you do electron microscopy, and this is a very poor picture, I should really replace it, but it basically shows very well that you can see the mitochondria and you can see the lipid droplets, and a really important aspect of this is that there's always or often a very close connection to the mitochondria and the lipid droplet, and I will get back to that later in my talk today. So why is this fat accumulation so interesting in the skeletal muscle? Well, the point is that if you have too much fat in the muscle, you might be in trouble. And this is one example of that, of a paper that we did now 2 years ago with the group in Austria, Rudy Zechner, where we basically study wild-type mice and mice with a knockout of ATGL. And ATGL is the main lipase in the heart and in the muscle and in adipose tissue. So it basically breaks down triglycerides to fatty acids. And if you knock out that gene, well, you can still store fat, but you can't break it down anymore. And what you will see in those animals is that they have massive fat accumulation. This is the heart, and this is at 12 week. Well, this is not lipid droplets anymore, but this is lipid Cs. It's really a big lipid storage. And those animals die approximately at 12 week. And not only because we sacrifice them, but also if you would Adipose, they would die. And they die from cardiomyopathy. So fat accumulation in the heart might lead to cardiomyopathy. A little bit less severe, but more related to the topic of this symposium here, is of course diabetes. There has been many studies in the late 90s and early this century showing that there is a negative correlation between the amount of fat that you accumulate in the muscle. That's always depicted on the x-axis here. So it's depicted as IMCL, and that's important to mention. When I talk about fat accumulation in the muscle, I talk about fat within the muscle cell. So really the lipid droplets in the muscle cell. And they are called intramuscular lipids, IMCL. So if you plot them on the x-axis and you have measures of insulin sensitivity, like the M value or the insulin sensitivity index, you will, in many studies, see a negative correlation between the amount of fat and insulin sensitivity of the muscle. So the more fat there is in the muscle, the more insulin resistant. These are our correlation, and so we did a study to see, okay, does this also happen in humans? So if we increase lipotoxicity or increase fat accumulation in the model, does that lead to insulin resistance? You can do that in several ways. You can give humans a high-fat diet, or you can give lipid infusions. And another model that we have used is fasting. Fasting is a very nice model because if you fast, your free fatty acids will go very rapidly in a kind of physiological condition. I mean, fasting used to be a physiological condition. I don't think that people fast much anymore, at least not in the Western society. So we did a study where we basically screened subjects and then had them fast for 60 hours or on a control diet, and then wash out and then crossover design, so they are their own controls. And again, we studied them in this respiration chamber so that we could study energy metabolism. But the other great advantage of the respiration chamber is that you basically can look up your subjects, so you're absolutely sure that they are fasted. Of course, I mean, they can always go out. We have an ethics committee in Maastricht, so they can always go out of the chamber if they really want, but then the experiment is failed. But I say this, but basically it is really important, because if you really want to have a fasting situation, then yeah, you need to be sure that that it's very well controlled. So we did it, and then we did clamps afterwards. And this is what you will see if you do fasting for 60 hours in humans. You'll see a massive increase in plasma-free fatty acids, but again, it's still physiological, so this is how humans can respond. Of course, you get a drop in glucose, and you get a drop in insulin, because you don't eat anything in the fasted state. But then we took muscle biopsies after the fasting or after the fat condition, and did the Oreo staining again. And what you can see is that you indeed have a massive accumulation of fat in the muscle. It doubles or even triples depending on which fiber you look. So for those who are not familiar with the muscle, muscle has basically 2 or 3 fibers types, type 1 and type 2. Type 1 is a more fat-oxydative fiber, and type 2 is a more glycolytic fiber. But anyway, you see fat accumulation in all fibers. So what happens then? Does this fat accumulation lead to insulin sensitivity or insulin resistance? And the answer is, well, very clearly yes. So these people have all been clamped in a hyperinsulinemic euglycemic clamp. So you basically give insulin, and then you adjust the amount of glucose. And the amount of glucose that you have to give with a certain amount of insulin is a measure of insulin sensitivity. And that you can calculate it as the glucose infusion rate. well, anyway, what you see is that they all develop insulin resistance. And if you calculate the average, then it's about a 50% reduction. And that is the same as what you will see in type 2 diabetic patients. So we made them as insulin resistant as type 2 diabetic patients. Again, I already mentioned we do have an ethics committee. If they eat again, they restore their insulin sensitivity again. So we are not creating our own type 2 diabetic patients. That's not really necessary, I think. So this is just temporarily, and this will go away again. Interesting was that what we also found in this study, we also looked at mitochondrial function, and we also saw that mitochondrial function was reduced in after fasting, which could indicate a kind of lipotoxic effect. And I'll come back to that later. So fat accumulation in the muscle, lipotoxic effects. So yeah, how does that work? Well, of course you could say fat accumulation in the muscle accumulates because you eat too much fat, so that is certainly one contribution. And of course, if you feed humans a high fat diet, they will store a lot of fat in the muscle, and it takes actually quite long in humans if you give them a high fat diet before they become insulin resistant. But the other point is of course the oxidation. And there are suggestions that maybe type 2 diabetic patients have reduced capacity to oxidize the fat in the muscle. So, is mitochondrial dysfunction the basis for lipotoxicity? And to measure this, as I said in the introduction, we use non-invasive and invasive methods. And one of the non-invasive methods that we use is magnetic resonance spectroscopy. And in this case, we measure 31 phosphorus. What you will do with this method is you basically measure ATP levels, phosphocreatine levels, and free phosphate. And we thought, okay, how can we carefully measure the mitochondrial function? Well, most likely we have to stress the muscle a little bit, so we do exercise in this scanner. And it's this kind of exercise, so the knee extension exercise, and then you measure the energy levels in the upper leg. So we made an exercise device for that, because this is of course a magnet, so you have to have something without any metal in it. But that worked, and now this is in place, and we use it quite often. And what we then basically measure is we focus on phosphocreatine. Phosphocreatine is an energy buffer in the cell, in the muscle, and if you start to do exercise, this phosphocreatine will be used to give ATP, and it will basically drop to a certain level, and then you will reach a plateau. Well, we're basically not very interested in that phase, but we are really interested in the recovery phase. As soon as you stop exercising, the cell tries to recover the phosphocreatine as soon as possible, because this is an important energy buffer, and of course the muscle doesn't know when he has to exercise again, so he tries to recover the phosphocreatine as fast as possible. And to recover this, you need to create ATP, of course, and for that you need your mitochondria. So it has been shown that a half-time of this recovery is a good measure of your muscle oxidative capacity. So the better your mitochondria are in your skeletal muscle, the faster this recovery goes. And as I said, with the same machinery, so the same magnet, you can also look at those intramyocellular fat. So basically this is what you do. This is the upper leg, with of course the subcutaneous fat, but also the interfibril fat, so that is really the fat that is within or around the muscle bundles, the muscle fibers, but we're not interested in that. We are interested, oh yeah, I should have known that. We are interested in the small voxels, and if you zoom in there, you get a kind of spectrum, and you can look at this intramyocellular lipid, and you can quantify that. So what did we find in those studies? So what we did is compare three groups of subjects. First of all, of course, type 2 diabetic patients, because we were interested in mitochondrial function in those patients, and we matched them with obese BMI and age mass controls. So all those subjects have a BMI over 30, they are around 60 years of age, but they are very well matched. And then we also studied a group of first-degree relatives. So you might know first-degree relatives are people that don't have diabetes yet, but they have diabetes in the family, and we know that they have an increased risk to develop diabetes. So we measured that, and basically what we found is that the type 2 diabetics, but also the first-degree relatives, had a reduced or a prolonged recovery time for the PCR recovery. So that basically means that the mitochondrial function is reduced. And so that is just the opposite. So it takes longer for the PCR to recover, so that the mitochondrial function is reduced. Or at least their oxidative capacity is reduced. Because of course we don't directly measure the mitochondria then, but we measure the muscle, and it could also be that the blood flow, for example, is disturbed. So because we are really interested in the mitochondria, we did in the same group also take muscle biopsies and focus on skeletal muscle mitochondrial function. So what we do is we make a small incision in the leg, you go in with a needle and you can take out a little bit of skeletal muscle biopsy, like 200 milligrams or so, and you separate basically the fibers, and these fibers you measure in a seahorse. You could do that, or we also, next to the seahorse, we also use this oxygraph system where you can do very nice experiments on mitochondrial function. And basically what we kind of traces that we do is are the following. So what you measure is oxygen concentration in the chamber where you put in your mitochondria, and from that you measure oxygen consumption. And what you do is you put in your mitochondria, nothing is really happening, then you put in the substrates, and the nice thing is you can play around with the substrate, so for example glutamate or fatty acid substrate, palmitoyl-CoA. Again, nothing is really happening because this is isolated mitochondria or permeabilized fibers, and they don't have endogenous substrates or ADP. But as soon as you add ADP, they will start to respire. And this respiration that you then measure, we call stage 3 respiration, which is basically basal mitochondrial respiration. Then we block ATPase by oligomyosin, and the respiration that you have left then is due to uncoupling. So if you have a high stage 4 respiration, it means that your mitochondria are uncoupled or are less efficient. Then we titrate FCCP, which is a gamma column coupler, which basically makes holes in the mitochondrial membrane, and your protein gradient will drop. The mitochondria doesn't like that, so he tries to build up this protein gradient again, and it starts to respire as hard as he can. And you get a high respiration, which is stage 2 respiration, or is basically the maximal respiration of your mitochondria. So with this way you can really separate where the defect in the mitochondria is, where it's in the coupling ratio, or in the maximal respiration or the basal respiration. So as I said, we did this in the same subject again. So in the subjects where we showed that in vivo mitochondrial function was reduced in the type 2 diabetics and in the first-degree relatives. And very nicely we saw exactly the same effect for stage 2 respiration. And also the order was the same, it's about 25% reduction. And for stage 3 respiration we see the same for the diabetics, and maybe the first degree are a little bit in between. And all these effects were independent of the amount of mitochondria. We have quantified the amount of mitochondria in different ways, with mitochondrial DNA copy number, or citrate synthase, or electron microscopy, but we didn't see any differences in the amount of mitochondria. So really the mitochondria per se were doing worse. So the conclusion from that study at that time, and of course others were doing these kind of studies also, is that diabetes is indeed associated with a lower mitochondrial function. This has been confirmed by other groups also in skeletal muscle. But it's an association, and doesn't tell you anything, of course, about cause or consequence. It could well be that this is just due to the diabetes. So we thought, okay, let's now try to improve this mitochondrial function and then see if we also improve insulin resistance by that. Because that gives you a little bit more clue for an idea what the role of this mitochondrial function in diabetes is. Well, and the simplest and easiest way to improve your mitochondrial function is obviously exercise training. So the first study we did on that topic is an endurance and strength training program in our lab, where we basically trained control subjects and type 2 diabetic patients, 60 years of age, match for age, match for BMI, BMI around 30, and kind of untrained, but I have to say that in the Netherlands it's really difficult to find really untrained subjects because we all cycle all the time. Have to, and the cars. So we followed them for 12 weeks and then we measured them after, and you can already see here from V2 max and V max that they indeed improved their aerobic capacity, so the training worked. To give you an idea about the training that we applied to those patients, so it's 12 weeks, measurements before and after, and they come in to the lab 3 times a week. One time they come in for resistance training, so we use all those nice machineries to train all the muscles in the body, and they do 8 exercises in the beginning with 1 set of repetitions, and later on in the training 2 sets of repetitions. And then 2 times a week they come in to do some exercise endurance training on a bike, but it's a relatively low intensity, so 55% of the maximum power output, which is not really, really high. They also have a warming up and a cooling down, so in total they were like 4 to 5 minutes on the bikes. So 3 times a week, I think maybe total 3 hours of training. All supervised and high compliance. I think the compliance was like 90%, they always came. So what happened to mitochondrial function? So again, and I now reverse so this is again this PCR recovery rate, but I reverse this now such that a lower level is a lower mitochondrial function. Again, if you compare controls and diabetics, and this is a new group, we confirmed that the type 2 diabetics have a reduced mitochondrial function, but then we see that training in both groups improves mitochondrial function. Well, that's nothing really new, of course. I mean, we know that training improves mitochondrial function, although it's important to see that type 2 diabetic patients are very well trainable, so they can also completely restore their mitochondrial function. This was mainly due to an increase in mitochondrial density, and actually it seems that the type 2 diabetics even have a little bit higher response, so they get more mitochondria, but we also did this oxygraph measurements again, and also the intrinsic mitochondrial function improved. So what happens to insulin sensitivity? So we did again the hyperinsulinemic euglycemic clamp with traces and so on, so you can measure muscle and hepatic insulin sensitivity. And of course what you will see is that the type 2 diabetics have a lower insulin sensitivity before training. You see that both groups improve, although in the controls there's only a very small improvement, but okay, they are not very insulin resistant also. The trained also improves. So you could say, okay, you increase your mitochondria and you increase your insulin sensitivity. You could also say, okay, they completely restore their mitochondrial function, but they only partially restore their insulin sensitivity. And I think that could be due to the duration. Maybe 12 weeks is not long enough to completely restore your insulin sensitivity. But we think that there's another component of the insulin sensitivity. Basically, you can divide your skeletal muscle insulin sensitivity, which is nothing else than taking up glucose after insulin, in 2 compounds. Glucose storage as glycogen and glucose oxidation. And we think that if you improve your mitochondria, you might improve your oxidative part, but this glycogen part, you might need other stimulus for that. And we are looking into that now, how you could do something synergistically to the exercise that we could further improve that. Hepatic insulin sensitivity also improved, but I have to be honest that this clamp, it's a high insulin clamp, is not the best way to measure insulin sensitivity. So we're repeating this study now, where we focus more on the liver, then we do 2 steps clamp, we also measure liver fat, that's a work in progress. So, okay, mitochondrial function improves, insulin sensitivity improves, still associated, of course. I mean, there's still no proof that then mitochondria are really important for your insulin sensitivity. So then we thought, okay, let's do another approach. Let's see if you have high mitochondrial function, if you're protected against diabetes. Well, we could do studies and wait for 20 years before people become diabetics, but we don't have so much time, so we did another approach. What we basically did is we selected students, trained and untrained subjects, so young subjects, normal BMI, again untrained, although 3.7 watt per kilogram is still quite good, but there's a marked difference between the trained and the untrained. The trained were really athletes. And then we thought, okay, we have to make them insulin resistant. As I showed you before, you could do this by fasting, but the other model that we apply quite a lot is lipid-induced insulin resistance. So basically what you do is you do a clamp in the subject, so you give them insulin in a human infusion, you give of course glucose to maintain the blood glucose levels constant, and you do this either with or without intralipid infusion. Intralipid is a triglyceride emulsion, you give some heparin to it, and then you basically have condition with high free fatty acids or low free fatty acids, and this is of course due to the insulin where you block lipolysis. So you get high free fatty acids, and let me go guide you through this. So the white bars are the untrained, the black are the trained. So if you first look at this graph, then you see that the insulin sensitivity is higher in the trained than in the untrained, nothing new. If you then look at this change, then you see that if you give lipid infusion, the insulin or the untrained subjects become markedly insulin resistant. There's a 72% reduction in their insulin sensitivity. But if you do the same in the trained subjects, this reduction in insulin sensitivity is much less. And this is even more clear if you look at insulin-stimulated glycogen storage. Again, maybe a little bit higher in trained than in untrained, but not a major difference. If you do lipid infusion in the untrained subjects, they become insulin resistant, but the trained subjects are completely protected against lipid-induced insulin resistance of the glycogen storage part. So we think that is really interesting, and most likely this indicates that in the trained subjects they are capable of storing the fat in the muscle in such a way that it doesn't interfere with insulin signaling. We also did insulin signaling in the study. We took biopsies and looked at that, and indeed partly confirmed that, although I have to say we took biopsies 5 hours after or during the clamp of 5 hours, and that's a little bit long to really have sensitive measure of insulin sensitivity. So we're repeating these studies now with a shorter duration. And this is just to show that the trained subject has high mitochondrial function, but of course this we knew. So what I've showed you so far is that mitochondria are reduced in type 2 diabetic patients. If you train, you improve your insulin sensitivity and your mitochondrial function. If you have high mitochondrial function, so if you are trained, you're protected, or at least partly protected against lipid-induced insulin resistance. So basically the message is all go out and do exercise, and then we'll save. And of course this we know, but this is not a message that a lot of people like. Not everyone likes to exercise, not everyone is able to do exercise. We also have to acknowledge that. So we're also looking for other ways to improve mitochondrial function. And some of you showed this picture today before, or a similar picture, where basically the effects of exercise in a very short way are depicted. So if you look at exercise and you see how it affects mitochondrial capacity, there's a central role of AMPK, of the NAD plus NADH ratio, maybe of SIRT1, although that's a little bit of a debate maybe, but also the effects on PTH1 are important. So basically exercise increases or activates AMPK, leads to activation of PTH1, and this leads to an increase in mitochondrial capacity. Calorie restriction basically does the same, and also leads to more mitochondria. But you've heard today already about resveratrol. Resveratrol is a compound in the food, in wine, but also in other products, that also can activate this pathway and might have exercise-like effects. So, Vern already introduced resveratrol. It's a polyphenolic compound. Its main dietary sources are grapes and wine and peanuts, and apparently it's very rich in the Japanese knotweed. No idea what it is, but it's very good. And there has been a lot of studies done in rodents with resveratrol, and like what you found, Vern, I think you will agree that basically most of those studies show positive effects of resveratrol. So it prevents weight gain in mice when they are on a high-fat diet. It improves biomarkers of metabolic syndrome like triglycerides, free fatty acids, glucose, insulin levels if you give resveratrol. It improves insulin sensitivity. The mechanism is most likely via activation of AMPK and SIRT1 and via PTH1 induced increase in mitochondrial number. And if you summarize all animal studies, you could say that resveratrol is capable of shifting the metabolic phenotype of mice when they are on a high calorie diet towards the phenotype of mice on a chow diet. So very potent results for resveratrol. So that made us very enthusiastic to do a human clinical study, or a proof of principle study, I would say. It's a small experimental study in which we wanted to investigate resveratrol. So basically what we did is a double-blind crossover placebo-controlled design with 30 days of placebo or 30 days of resveratrol, then a washout period of 4 to 6 weeks, and then all subjects underwent the other arm, so they are their own controls, and we did a lot of measurements during those 30 days. We gave subjects 150 milligrams of resveratrol in 2 dosages per day, so 75 milligrams in the morning and at dinner. And for those who don't like to take pills, you have to drink 10 liters of red wine to get the same. So, and I'm not sure if that's advisable. Although it might be fun. So these are the subject characteristics. We focused on in first instance on a subject group of obese people that are at risk of developing diabetes or are insulin resistant, but are still fairly healthy. As you can see from the glucose and also the blood pressure, it's not. They're quite healthy actually. So we followed them and we gave them resveratrol for 30 days, and these are just the blood safety parameters after 30 days. Well, there were no any safety issue. All the markers, no major effects, not negative, and also no positive effects on these markers. Although there was a tendency, and I'll come back to that later, for a reduction in ALAT, which is an enzyme of most likely reflecting liver fat accumulation. So we looked also a little bit more at the metabolic parameters. So of course we were interested in glucose and insulin, and we noticed that there was a tendency or significant reductions in plasma glucose and insulin. They are all small changes, but they are very consistent. As a result, the HOMA index is of course improved. which could indicate that the insulin sensitivity is improved. I'm very careful here because we didn't do claims, so I can't really say whether insulin sensitivity improved, but I mean, they go into the right direction. The same with triglycerides, they also improve, also go down, and some inflammatory markers also improved after 30 days of resveratrol. And again, these are, the subjects are their own controls, so we could really compare that. We measured them for 36 hours in these respiration chambers, and I showed you before, because it has been suggested that resveratrol would also affect energy metabolism, and of course because it affects mitochondria, you can expect that it does something to energy metabolism. The nice thing about these chambers is that you can measure energy metabolism during 24 hours, but specifically interesting is the night, because during the night you can very reliably measure someone's resting energy metabolism. You don't have interference of activity or of the diet and so on. And it has been shown that if you have compounds that affect energy metabolism, you can best measure them during the night. And that's also what we found. If we measure sleep and metabolic rate in placebo or resveratrol, we saw a significant, again, small, it's human research, you have to get used to small differences, but a small and consistent reduction in sleep and metabolic rate after resveratrol. Is this a good or bad thing? If you want to lose weight, it's most likely not a good thing because you want to have high energy expenditure. On the other end, if you start to do training, you also will see that your sleep and metabolic rate goes down, and it could be a sign of more efficient mitochondria. So I think it's a sign of a better metabolic health. No effects on body weight, also no effects on 24-hour energy expenditure, and that is because you get too much interference with physical activity, and it's not sensitive enough to pick that up, such small differences. Then we did microarray and skeletal muscle biopsies that we took after the treatments, and we compared it to animal data. Animals, mice that were treated with resveratrol, and basically the conclusion was that we see an upregulation of genes that are involved in mitochondrial metabolism and a downregulation of genes that are involved in inflammatory pathways. So then, of course, we were also interested, okay, does resveratrol activate the pathway that you expect from animal research? And indeed, we could show that after 30 days of resveratrol, we got increased phosphorylation of AMPK, which is a measure of AMPK activity. We got increased SIRT1 at the protein level and increased PGM1 at the protein level, all indicating that indeed also in humans this mechanism can be activated by resveratrol. So then, of course, we also looked at mitochondrial metabolism, and there again, most of the data confirm that resveratrol improves mitochondrial function. So we see an increase in citrate synthase activity, no change in mitochondrial number, so the amount of mitochondria doesn't seem to be changed, and also no effect on this PCR recovery measure, but again, that might also be not very sensitive to pick up differences. So that's why we also did the mitochondrial respiration measure in the oxygraph, and then we could see that especially when we had octanoate as a substrate to the fibers, we see an increase in mitochondrial respiration, both in state 3 as in state uncoupled. So all these effects were very consistent with each other, showing improved mitochondrial metabolism. We also took adipose tissue biopsies. This has recently been published now in another paper, and basically what we saw is that resveratrol changed the adipose tissue size, that you get smaller adipose sites and a shift in the distribution, so less bigger and more smaller adipose sites. And we also did some areas on that, and we also saw improved markers of inflammation in the adipose tissue. So of course we were interested in the ectopic lipid accumulation, so we measured liver fat with magnetic resonance spectroscopy, and here you see just two examples. Those subjects didn't have a lot of liver fat, and I discussed that yesterday with some people. It's apparently in the Netherlands, it's very hard to find people with fatty liver. We don't know why that is, but it's really difficult. But anyway, there was a significant reduction in the amount of liver fat. Basically, all subjects went down. Not markedly, small effects again, but very consistent. However, one of the most striking and for me really exciting findings is what happens in the skeletal muscle. So we did again these already stainings, double stained them with fiber type, and we looked at the amount of fat in the muscle. And then what we found is that after 30 days of resveratrol, you get more fat in the skeletal muscle. It is a very marked effect. I've actually never seen such a clear effect of such a small intervention. It's just taking a pill for 30 days, and you see so much fat accumulation in the muscle. So the question is, of course, is this now good or bad? Because I started my talk with saying, okay, fat in the fat accumulation in the muscle is not really a good thing. Skip this one. And I go immediately to this one and explaining to you something that is called the athlete's paradox. And what I showed you in the beginning of my presentation is basically this data, those three columns. And it is that if you measure the amount of fat in the muscle and you go from insulin sensitive to insulin resistant people, that there is an inverse correlation. So the more fat there is in the muscle, the more insulin resistant you are. However, if you measure endurance trained athletes, they also have a lot of fat in their skeletal muscle, and of course they are very healthy and they are very insulin sensitive. So that is a paradox. How do we explain this? And then we thought, okay, let's see how this fat accumulation in the muscle starts. Why does exercise lead to fat accumulation in the muscle, and why does resveratrol lead to fat accumulation in the muscle? And then if you look at the pathways, as I showed you before, they all share PHC1. So both exercise and resveratrol lead to an activation of PHC1. And this PHC1 leads to the activation of mitochondrial metabolism, but maybe also to muscle fat storage. So together with that Muyo in Duke, we did a study on PHC1 transgenic animals. And well, to make a long story short, basically what we see is if you overexpress PHC1, you indeed get more mitochondria in these animals, this is known, but they also store much more fat in their skeletal muscles. As you can see there, very large lipid droplets, nice mitochondria also, and very nicely aligned. So most likely good lipid droplets close to the mitochondria. We did the same, did this as whole body, so you can get all kinds of adaptations. So we also used human primary myotubes and overexpressed PHC1 in those muscle cells. And basically we found the same. If you overexpress PHC1 in those myotubes, you get more triacylglycerol accumulation in those tissues. So then we looked at some gene expression of genes involved in lipid metabolism, and one of the major genes that was upregulated, I mean a lot were upregulated, DGAT and ATGL, but one of the major one was also PLIN5. That was a gene that really caught our eye and thought, okay, that's interesting, and I'll come back to that in a second. So then we thought, let's go back to the muscles of the trained and the untrained subject and see what's happening there. So we got these samples from the trained and untrained subjects. Yeah, that's correct. The white is now the trained subjects. This is the amount of fat. So you indeed see that trained subjects have more fat in their skeletal muscle. We did some mRNA expression, and you see some fat oxidative genes that are higher in the trained subjects. But again, also this PLIN5 was higher. And also if you do Western blot and you look at those PLIN2 and PLIN5, they are increased in trained subjects. So what are PLINs? PLINs are paralipins, and they basically cover your lipid droplet. So the lipid droplet in the muscle is covered with all kind of lipid droplet coating proteins that basically regulate the lipolysis of the lipid droplet in the muscle. So then we thought, okay, maybe if you have a lot of those splints, you can store your fat in the proper way in your skeletal muscle, and maybe this might reduce lipotoxicity. So we first focused on PLIN2. So in the muscle you have basically PLIN2 and PLIN5 that are the major lipid droplet coating proteins. So we first did an approach where we overexpress PLIN2 in skeletal muscle cells. This is C2C12 cells, so a mouse cell line. Overexpress PLIN2, and I'll go a little bit fast through the data now. Basically what you see here is if you overexpress PLIN2, you get more fat accumulation in the muscle, you get more incorporation of your fatty acids into triacylglycerol, but also into diacylglycerol. But then basically this is the major data. If you then look at insulin sensitivity of those cells, this is the insulin sensitivity of your control cells. If you give palmitate to the cells, you induce insulin resistance. But if you overexpress PLIN2 and you give again palmitate, you don't induce insulin resistance at all in these cells. So those cells are completely protected against lipid-induced insulin resistance. Okay, that's cell work, that's nice, but we're always interested in the in vivo situation. We are not very good in making transgenic animals in Maastricht. We don't really have the facilities, but we use a lot this technique, and that's a very nice technique if you want to study muscle physiology. And basically what it is in vivo DNA electroporation. What you do is you take an animal, a rat normally, and you inject in one leg a construct with your DNA of interest, behind the promoter of course, so that it's transcribed, and you give a kind of electric pulse over the muscle. This makes the muscle cell membrane permeable, the DNA goes in, you let the animal recover, he's back on his feet in 5 minutes, and then you wait for like 3 weeks or so, and you will get overexpression of your gene of interest in this leg. And the nice thing is of course that you can do one leg with your gene of interest and the other leg as a control with another vector or another gene, whatever you want. And then we do clamps in those animals, so we do surgery on them, put in the cannulas and can clamp them. So what you will get is this model. You have one rat but with 2 different legs. So this is the normal leg, which doesn't overexpress PLIN2. This is the other leg that has overexpression of PLIN2, and as a result, one leg will have almost no fat in the muscle, and the other leg has a lot of fat in the muscle. So within one rat, all the system circulation is the same, so there's really only a local overexpression in your muscle. So we did this in this animals with PLIN2, and I just showed you already that it leads to more fat accumulation, and you can clearly see this here. This is the normal leg without PLIN overexpression, and this is the PLIN2 overexpression leg, and you see a lot of those lipid droplets very nicely located there. Then if we measure insulin sensitivity, and this is muscle-specific insulin sensitivity, you will see that this was on a chow and on a high-fat diet, that in both conditions, if anything, the insulin sensitivity is increased. It's not majorly increased, or you can even doubt that, but it's not reduced, and that's the message, I think. So despite that they have a lot of fat accumulation in the muscle, so ADRP is PLIN2, by the way, that's the same name for another protein. So despite the fact that they have a lot of fat in the muscle, they are not insulin resistant. So that mimics very well the athlete's paradox. Well, I have to hurry up a little bit. Yeah. So we basically have the same results with PLIN5. So you just have to believe me, otherwise look it up. But it's exactly the same data. So also more fat accumulation and also no insulin resistance. So this is the model that we proposed from these studies, and basically we think that exercise, but also resveratrol, activates PHC1 and AMPK, but it's a simplified figure, that leads to expression of target genes, which will increase mitochondrial biogenesis, but at the same time it also activates a lipid droplet gene program, which leads to the expression of perilipids, but also ATGL and other lipid droplet coating proteins. And why is that regulated in this sense? Well, for two reasons. This lipid droplet is important because it can deliver fatty acids to the mitochondria, and exercise, you increase your mitochondria, of course, because you're supposed to do exercise. The body thinks you're going to do exercise later on, and then you need your lipid droplets as a fuel for your mitochondria. And I'm still allowed 3 minutes or something.
Oh, you're at that. Well, it's Vern's show.
Sorry?
Vern, 3 minutes. Catch your point.
The other nice aspect, I think, is that there's now recent data that the free fatty acids coming from this lipid droplet not only serve as a fuel, but they might actually also activate this PH1, so you get a kind of feedback loop. So, is that relevant for humans? And to answer that study, or maybe partly answer that study, I want to focus on this last aspect that I said, and that is that the free fatty acids coming from this lipid droplet can activate this PH1 or other genes and lead to the expression of oxidative genes and thereby stimulate mitochondrial oxidation. So you get a kind of circle loop. And as I said in the beginning, ATGL is the main lipase responsible for the release of free fatty acids. So I showed you one of the first slides that if you knock out ATGL, you get this massive fat accumulation in the muscle, and I told you that those animals develop cardiomyopathy. And we measured in those animals mitochondrial function, and then we could show that if you compare wild type with a knockout, that indeed all kinds of genes involved in mitochondrial metabolism are reduced. But then we found out that this is basically not due to lipotoxicity, but this is due to the fact that those animals, those knockout animals, can't release the free fatty acids that are necessary to activate PH1. And we could show that by giving those animals a PPAR ligand, pharmacological PPAR ligand. And if you do that, you could completely restore the reduction in mitochondrial gene expression. And the same is true for mitochondrial respiration. So wild type knockout, they have low mitochondrial function. By giving them PPAR agonists, you can completely restore them. Because basically you bypass the free fatty acid, you just give another ligand for the PPAR activation. So why is it important for humans, we believe? Because there's also humans with mutations in ATGL. And those people have NLSDM, which is a disease called neutral lipid storage disease with myopathy. And those patients, they store a lot of fat in their muscle, in their heart. 50% of the patients, it's a rare disease, there's only a few patients, but 50% of them die at a very young age, like 30, 35 years from cardiomyopathy. And the others have exercise intolerance problems, they have myopathy, so they have a lot of problems. And there's no cure for those patients. We were able to study 2 of those patients. As I said, it's a very rare disease. There's only like 20 to 30 cases in the world. We were able to study 2. And of course, based on our animal data, we thought, okay, let's give them a PPAR agonist, so let's treat them with fibrates. So we did that for 28 weeks. We measured all kinds of measures of the muscle performance. So one up here is the leg extension test, so muscle strength test. And these are the 2 patients. This is the control values. You can see that they are way down, so they are very low muscle strength. But with the treatment, they improve. This patient we only could measure until 12 weeks. We measured her on 28 weeks for some other parameters, but she was so ill that she didn't want to do this test. It was too exhaustive for her. We measured insulin sensitivity. We don't have control values here, but we could show that The metabolic flexibility, which is basically a kind of side mark for mitochondrial function, improved after treatment. We took muscle biopsies in those patients and we measured fatty acid oxidative capacity, and again we could see in both patients that the beta fibrate improved their mitochondrial function. This is the control value, so they almost restored to control values, or actually patient 2 completely restored to control values. So the main problem in those patients is fat accumulation in the heart, in the muscle, and in the liver. So we measured fat accumulation in those tissues. Patient 1, patient 2 in the control. Well, here you can already see from the yellowish picture that there's a lot of fat in the muscle. They have 20-fold more fat than any other patients we will ever see. But once we started treating them, this fat accumulation really reduced. And if you calculate it, it's a 50% reduction. They're still much higher than normal subjects, but the fat accumulation reduced by 50%. And you can see the same in the heart. This is the heart fat, again the control value. So they have much more fat in the heart, but after treatment they start to decline. And for the liver, it was less clear. There seems to be a reduction, but then it seems to restore again. So it seems to be beneficial for those patients. We also did heart function that improved maybe slightly, but we think that the duration was just too long. So those patients are still on fibrates, and yeah, we will follow them up in the future. So that leaves me with the final slide. Sorry that I went over the time, but it's a little bit an open question, I think. I think if we, or an open conclusion, if we study lipotoxicity in the muscle, there's many players that play a role. It's not so easy as we thought a few years ago. Oxidation seems to play a role, APK, exercise, but also the storage capacity of the muscle. And in the end, I think our conclusion is that, well, we study the muscle because we think the muscle is important, not only for contraction, because also it plays an important role in insulin sensitivity. The muscle takes up 80% of the glucose after a meal, and we think that a healthy muscle is a muscle where you have a good balance between oxidative capacity, the amount of fat, but also the lipid droplet dynamics. So the way you store the fat in the muscle seems to be very important. But I think this is now really an open field, and we need to do much more studies on how these lipid droplets are connected to the mitochondria, what the role is of the size of the lipid droplet, and so on and so on. So that's what we will do in the future. And with that, I would like to stop, but of course not without thanking my research team that we built up in Maastricht. I showed you data from Sylvie Temmers, who was doing the Resveratrol studies, but she's now doing Resveratrol studies in diabetic patients. So we're following that up. I showed a lot of data of Madeleine Bosma, who's now in Karolinska, but who did the lipid droplet protein work. And I had this group together with my colleague Matthijs Hesseling, and my wife, Viv, is responsible for the magnetic resonance spectroscopy studies in our group. So I think overall we have a very nice team, and of course there's a lot of people with whom we collaborate. I showed you data from Deb Muyo, from Rudy Zechner, Johan Auer we collaborate a lot with on the role of Resveratrol and all the pathways in there. And of course, thank you for your attention.
So I'm sure Dr. Schrauwen would entertain some questions. Dr. Gordon.
Thank you for a very nice talk. The question that struck me right away was the intramuscular triglyceride stores compared to type 2 diabetes in athletes. I'm just wondering if glycogen stores parallel that, or if that's very different.
No, yeah, I think that's different. I'm not sure if people really consistently looked at that, but of course the patients have a lot of glycogen in their muscles. That's a good one. I think it's not the same paradox.
What's the effect of the insulin on the regulation of the
So insulin.
Insulin effect on the blends. Is there no? Is it regulated by insulin?
There's data coming out now that it is indeed regulated by insulin. They're suggesting that they also might be activated, phosphorylated in these kind of things. But there's not a lot of data known yet. The paralympics, I mean, they've been studied more in adipose tissue, and in the muscle it's really a new field, and also in the liver they find similar effects. But yeah, it's a good question. Yeah.
It's one of the things that strikes me about one thing that's similar between type 2 diabetes and athletes is actually they have low insulin levels, right? They're actually insulin deficient in a sense.
And not only because I mean the patients could have said it maybe, but the patients that we study are quite often early diagnosed type 2 diabetic patients. They don't use insulin, so
That's my point. They have low insulin, as
Oh,
do the athletes.
they are hyperinsulinic. They are in the early phase that we study them. So
They shouldn't be hyperinsulinic if they're actually hyperglycemic already. I mean, so they should be relatively hypoinsulinemic at that point, which may be like the athletes.
yeah, yeah, okay. Yeah, so you wonder about the effect of insulin on I don't know. We should study that. I don't think it has been studied. No.
Just further that a little bit too, maybe I missed it, but what's known about the effect of resveratrol on the planes or ATGO activity / expression?
No, I didn't put in that data. That's work in progress, but we've shown that, I mean, in those humans where we treated with resveratrol, they also increase the paralipids, and now we're doing studies in human primary myotubes. We give them we give the cells resveratrol, and then we also see this increase in fat accumulation in the muscle. So it really seems to be a direct effect on the muscle, and we also see the induction of the paralipids. And I don't really think that this PH1 plays an important role in there. But of course we should prove that by doing PH1 knockout or something. I don't think we will go along that way. That would be the proof, of course.
So Patrick, are there any differences in subcellular localization of the lipid droplets in the trained athlete versus the type 2 diabetic?
Yeah, that's really an important question, I think, and that's absolutely, yeah. So you have the lipid droplets more around the subcellular level mitochondria, which are and they might be more interfering with insulin signaling, or at least in the diabetic. And then you have the intermyofibrillar mitochondria, and also there the lipid droplets. But there's only data from 1980s from Hans Hopper in Switzerland. He did a little bit of electron microscopy and compared or looked at the lipid droplet and contact with the mitochondria, but all in the training, and not really in diabetes. So we are currently doing that, and trying to see what is the lipid droplet size, where are they located, how far are they from the membrane, and does that relate to insulin sensitivity? So we're doing it now. Yeah, that data is not out yet.
That was fantastic. So many great parallels with things we're interested in here as well. Until your last talk, few slides on neutral lipid storage disease, I was going to argue that high levels of free fatty acid oxidation are the actual cause. Like, we measure fat in the tissue, but we're not measuring the amount of fat oxidation at the same time. And there's a few reasons for that, that I think that. Patients with mitochondrial myopathies that we studied at Southwestern, we fly them in. Their VO2s are 15. We have significant exercise deficiency. None of them have diabetes.
Yeah.
They have a lot of fat stored, but I think it's the either dysregulated, like Dev Munoz, dysregulated mitochondrial oxidation and acetylcarnitine profiles that may be the actual culprit. And what we're just able to measure is the fat that's stored there. And there's some great data coming out of Dan Kelly's lab where when given fibrates to, I think they weren't ATGL knockouts, but they had overexpressed cardiac specific steatosis, giving them fibrates worsened their cardiac function.
Okay.
So I've always gone back and forth, and the one thing that athletes have is that they can upregulate that fat oxidation, but it's in a balance, right?
No, I think that the Muyo hypothesis is very interesting and very attractive, and together with her we're also studying it now in and we want to do carnitine supplementation and these kind of stuff. Because I agree. I mean, I think that has been a little bit ignored. People look at the fat accumulation, but the fat accumulation will not completely answer the question or give an answer.
Sort of like
and MSDM is, I think, a little bit different story. I mean, that's really a particular disease, of course. So they really have problems in releasing fatty acids.
Yeah, no one brings up the old Randall cycle.
We do.
You do.
No
one talks about it, but it's been around forever, right?
Yeah.
How is fat are going to suppress glucose oxidation?
Absolutely, I completely agree. And in the diabetes paper on the trained and the untrained with lipid-induced intramuscular, we bring that up. We say, well, most likely it's just a Resveratrol cycle that's operating there that could explain completely the results. And we have another paper in PNS where we block CBT1, so block fat oxidation, and then you get. I mean, you basically also have a Randall cycle-like effect, and they also become more insulin sensitive. So I agree completely with you. I think it has been ignored too long, and it's good to have a look at that again.
And your fasting data.
Yeah,
Fasting data also support that.
could also support it. Yeah, absolutely.
Okay, so first Dr. Redpoll, then Dr. Fernando.
Okay, I'm a little bit in kindergarten here, but I would assume that accumulation of fat in skeletal muscle would somehow correlate with accumulation of fat in cardiac muscle. And I was wondering within the Resveratrol if there's a dissociation there, or maybe it's not, it's not correlative. But as you were continuing, you did show that slide where you were seeing some sort of associations in your last subject groups. But if you could speak to that, and also, is this then, because we're not seeing a change in body weight with Resveratrol, and could it also be then part of this redistribution of the fat?
Yeah, so the first question, I mean, we didn't look at cardiac fat in the Resveratrol trial, and we're doing that now. So we can see that maybe in animals you can have a look, I don't know, or you maybe did already. But we know that there is not such a thing as an athlete's paradox in the heart. So if you measure fat accumulation in the heart in athletes, it's very low. So it's not the same as in the skeletal muscle. So I think that's actually very interesting, because that must mean that there is other ways in which the fat is stored in the heart than in the muscle, and also it has other functions maybe. And the other question was about body weight and Resveratrol.
Well, there isn't a change with body weight in the treatment of Resveratrol, so what it appears, at least from my understanding, is that what might be going on is there's a redistribution of the fat.
Yeah, that could be. I mean, we did 30 days, which is too short to really have a look at it. So we want to do, now we're starting that up a 6-month Resveratrol trial, and then we will also look at body composition and again, fat accumulation in the tissues, and then see if there's maybe a redistribution of the fat. But I think we need longer trials also to see if there's an effect on body weight, because 30 days is too short, I think, to say anything about.
Well, you're not supposed to lose that much weight on a 33,000.
No, so that's why we want to go longer and see if there's what is the effect is, because we do have this reduction in sleep and metabolic rate, which is not necessarily a good thing, from at least not from an obesity perspective. So that might be worrisome. So we need to do a long-term study, I think, to see what happens if you give it for 6 months. Do you gain weight, or do you just compensate in another way for that? And what happens to the body fat composition in the end?
Do you notice any difference in the ultrastructure of the mitochondria that are associated with the droplets versus unattached mitochondria?
Yeah, there have been studies. I mean, like Dave Kelly has done studies showing that in type 2 diabetic patients, the mitochondria might be smaller, they might be sometimes more damaged. But there's not so many studies out there. Electron microscopy is a lot of work, of course, so it's not so easy to do that. We also did a study with electron microscopy in those groups, and we basically confirmed that the mitochondria are a little bit smaller in type 2 diabetic patients, but we didn't compare trained there. But
it could well be. Picture where you had a droplet with a big mitochondria. Is that common? I mean, are these things acting like foci for mitochondria?
mitochondria binding to the droplets.
Yeah, so we have shown for this PLIN5 in another paper that this PLIN5, so this lipid droplet coating protein, you can also, we can detect that in mitochondria. We can see it if we do immunogold, we see it on the mitochondria. If we do isolated mitochondria, we see it in the mitochondria. So we think that this protein connects the lipid droplet to the mitochondria in a way. Why, we don't really know, but so there seems to be really an interaction. And that are the kind of stuff that we want to do now. Measure the interaction size between mitochondria and lipid droplet and see if that's altered or different in type 2 diabetic patients. Yeah.
Okay, so just before we break, I would like to take the opportunity to present Dr. Schrauwen with this bag of goodies as kind of a token of our appreciation for coming all the way from Europe for our symposium. We really appreciate it and your participation in our symposium. So I also like to thank all of the organizers and the other speakers in today's symposium. So thank you very much for staying to the end as well.
If you